Hello everyone, I am David Bonnell, a Senior Vice President at Aliri. Very pleased to be with you today and with my colleague, Dr. Corinne Ramos. The objective today is really to speak together during this webinar about our way and the way we are building a smart biomarker strategy at Aliri. So thank you very much for joining us. Extremely pleased to be with you. And we will start now. So what can you expect from this webinar today? So we will cover 4 different topics. The first one will be why we see so many failures during, let's say, clinical trials, more particularly. The second part will be really dedicated to the mechanism-driven approach that is becoming really essential in our, I would say, industry today, more particularly when working on translational and clinical biomarker projects and studies. Corinne will share with us how to build a smart biomarker strategy, and Corinne will use different examples, study cases to illustrate this smart approach. And as you will see, we will also discuss about practical insight to improve translational decision making. So at the end of this webinar, we will have a Q&A session. Feel really free to ask any question you may have in the Q&A box available on the interface of the webinar, and we will take a few minutes at the end to answer your questions. So, why do so many promising drugs fail in clinical development? So today, one of the main challenge the pharma biotech industry is facing is really the attrition rate. So challenges associated to the, let's say, drug candidates, and 90% of the drug are failing during clinical development. And if we look at the last data associated to these failures, we can see that around 40–50% of this failure are due to a lack of clinical efficacy. So this topic, the efficacy, is really extremely important today. And we can also see a lot of challenges associated to the translational gap. So jumping from the preclinical models to the clinical outcomes is today and since several years extremely challenging. So it is also one of the reasons why so many promising drugs are failing. An important topic, and you have understood this already in my previous slides, this is the evidence that will make the link between mechanism of action and the clinical outcome. So in few words, the efficacy of the treatment. And this is why the biomarkers are so important in order to be able to confirm this efficacy. So the readout biomarkers that we can follow from preclinical to clinical stages are usually added too late to the programs, or the strategy is not well defined or not clear enough. So this is why we see a so huge attrition rate right now in our market. So if we focus a little bit just on the biomarker part, of course, we are using the biomarker analysis from discovery to clinical stages, as you may know. So in discovery, usually it's mainly associated to the target validation. During preclinical stages, the objective is really to identify, confirm the mechanism of action by identifying or by targeting some biomarkers directly in animal models, for example. The early clinical phases are more dedicated to the target engagement directly in patients' tissues when available, and also to the pharmacodynamics effect of the treatment. And during clinical development, of course, the main focus is really on the drug efficacy. So in few words, the mechanism of action. An important topic is also associated to the patient stratification. Thanks to all the tools we have today and all the AI data science approaches, it's today possible to characterize the phenotypes of patients to really be able to identify, for example, a resistance associated to a specific phenotypes or the efficacy, of course. As you can see on this slide, we have all to keep in mind that one of the main challenges really is the translational gap when working on biomarker. Being able really to identify this biomarker during the preclinical stages and jump, let's say, on the human tissues in order to confirm again the efficacy based on the mechanism of action, for example. What we can observe more and more, this is really a regulatory shift. Um, Pretty recently, the FDA, for example, emphasizes the mechanism-based evidence. A very nice draft guidance is available on the FDA website. And very recently, two KOL, key opinion leader from the FDA, published in the New England Journal of Medicine one scientific article promoting the use of a single well-controlled clinical trial. The objective is, of course, to accelerate the clinical development. And this type of, let's say, shift is really associated to a high quality mechanism linked evidence. So in few words, we have really to confirm the efficacy, confirm the mechanism of action to be able to just use one single well-controlled clinical trials. For now, it is just a draft guidance from the FDA, but we can see that, let's say, it's certainly the future of what we are doing. The experts consider the biomarker and the mechanistic evidence as part of the confirmatory evidence. And the mechanistic and the pharmacodynamics in this document are explicitly mentioned as valuable evidence types. So more than before, the biomarker are key in order to confirm again the efficacy and of, excuse me, any drug directly in human clinical trials. So this is why having a smart strategy is key today to really to improve, optimize the time and the money invested on these studies. At Aleri, few words about Aleri. Aleri is a CRO, a contract research organization. And what we are offering is really quite unique. We have the capability to combine classical bioanalysis approaches with spatial biology and bioanalysis um, approaches. So in few words, we can work directly on fluids, tissue homogenate, as well as on solid tissues. So we can confirm that the drug is at the right place, at the right concentration, and really what is important today during our webinar, we can confirm the efficacy of the treatment based on some biomarker analysis. The name of the company is maybe new to you, but the company is coming from a merger of three main and famous CROs. I will come back on this topic. And thanks to this huge expertise, this important expertise we have, we can offer the bioanalysis, spatial bioanalysis, and spatial omics approaches, as you can see on this graph, in order to focus on different topics. And the mechanism of action is really one of the most important type of services we are offering, being able to work directly on this biomarker during preclinical, translational, and clinical stages. As I mentioned before, the company is coming from the merger of Pixent, IMA Biotech, and Tandem Labs. We have today 3 sites, 2 in the USA, 1 in Europe, 30 years of experience and expertise. So the Salt Lake City site is 30 years old, the Colorado Springs site is 25 years old, and the French site is 15 years old. So several years of, let's say, biomarker analysis expertise dedicated to our clients and partners. And as I mentioned before, we have the possibility to work directly from fluids and bulk samples by using our LC-MS/MS platform, more than 40 instruments dedicated to this. But we are also offering qPCR, mass cytometry, NGS analysis directly on this type of, let's say, tissues. And what is quite unique in the CRO world, this is our capability to also analyze some solid tissues by working on tissue section to preserve the tissue microenvironment. And we are using different platforms at Aleri, like the classical digital pathology platform, as well as the quantitative mass spec imaging, the imaging mass cytometry, the genomics, the Visium. So in few words, we have several platforms available for our clients in order to target directly any biomarker you will see in the tissue microenvironment. Something quite unique as well, we have the wet labs. So we have all the platforms on our sites, and we have also the data science team that will use artificial intelligence, deep learning, machine learning, if needed, on the project with our clients in order to combine the results coming from all these platforms. So today, we will really show you how we are creating a smart strategy based on this huge expertise available at Aleri. And my colleague, so Dr. Corinne Ramos, will jump here. So Corinne is one of our subject matter experts, more than 25 years of experience and expertise in the biomarker assay development field. Corinne is today R&D director involved in a lot of projects with the operation, excuse me. Corinne has published a lot in this field. So Corinne, I'm very pleased to let you share with us what is a smart strategy at Aleri.
So thank you very much, David, for the nice introduction. So before I'm getting into the detail of the smart strategy for biomarker development, I wanted to give a little bit of rationale behind it. So the modern therapy, as you all know, does not act in isolation. They act within the complex tissue ecosystem where the treatment response depend on the multiple biological layer. Like, for example, the target engagement, pathway activation, in some case, the immune context, and as well the microenvironment remodeling, the cell-cell interaction. And yet we have a lot of biomarker strategy right now that still rely on very fragmented assays that measure totally isolated signal. And you can see just an expression level that you can measure, like with a bulk, like assay for biomarker, rather than measuring the underlying biological mechanism of the therapy. And that gap, which is totally created, it's between what we can measure and what do we need actually, like to measure to understand the biological relevance of the therapy. And we can close that gap now with having a successful biomarker strategy that needs to be grounded in biological mechanism. And now, like how did we design a biomarker strategy that actually follow the biological mechanism of the drug? This is my next slide. So mechanism-driven biomarker strategy follow the biological cascade from drug exposure to clinical response. And each step of this biological cascade generate measurable signal that can help understand why a therapy is working and why a therapy is not working on certain patient. So here an example of one therapy, which is a checkpoint inhibitor, an anti-PD-1. So you see that cascade where the drug is going actually, like to bind to the receptor, the PD-1 receptor. So you can start measuring, like the PD-1, the receptor occupancy, as well, that the expression of the PD-L1 in that particular example. And that will lead, like to pathway modulation that need as well, like to be measured. And this pathway modulation, which can involve an interferon gamma signaling, will induce, like a cellular response, like, for example, a CD8 activation, a granzyme B expression, which in turn will lead, like to remodeling the tissue architecture with more abundance or infiltration of new cells that will lead actually, like to eventually a clinical outcome. In that case, it could be a tumor regression or durable response. So the challenge here, it's become, how can we translate this biological cascade into a very robust biomarker strategy? And this is why we are using a smart strategy for biomarker development here. So the biomarker strategy needs to be specific, linked to defined biological mechanism. It needs as well to be measurable, quantified with robust and reproducible assay, achievable as well within the constraint of the clinical sample analysis and the constraint of the clinical trial as well, relevant, That means that it needs to be connected to the drug mechanism and the disease biology, but most of all, relevant for clinical decision during the drug development effort. And finally, time-bound. Data available needs to be early enough to inform decision in the program. So I'm going to walk you through all the different elements of this SMART strategy. And the first requirement is specificity. The biomarker strategy must reflect the biological mechanism of the therapy. So the mechanism-driven biomarker, as I already told you, are totally anchored in biological hypotheses. And here I'm showing an example. For example, the KRAS inhibitor is going to inhibit the MAP kinase pathway. And one of the readouts of this inhibition is going to be the phospho-ERK, which is at the end of this signaling pathway. And that, readout, it's going to be a mechanism-driven biomarker. The same way for a TGF beta inhibitor, the biology is going to, for that particular inhibition, it's going to be a stromal remodeling and fibroblast activation. Those are going to be the readout, actually, of the predictivity, of the efficacy of the therapy. And above this, and this is the example that I'm showing on the right side on the right side of this slide. Above this, it's not only about looking at just the abundance or the localization of an expression, but it's as well looking at the proximity between the different marker. And in that particular case, we're not showing just a CD8 infiltration, but we are showing the proximity between the CD8 and the tumor in that particular instance. So this principle applied to more complex modality. And let's talk about the mechanism-specific biomarker for antibody drug conjugated, for example. The biological mechanism of this ADC rely on different biological layers. There is, the target binding. There is after the internalization of the antibody and as well the cytotoxic payload release. And many ADCs payload induce the DNA damage in tumor cells. And one of the readouts, actually, of this DNA damage is to look at a marker, which is the phosphorylated H2AX, which is a marker of DNA double-strand break. So this biomarker is becoming the new mechanism-driven readout, actually, of the efficacy of your treatment. So you have an interpretation and the quantification of this marker on the right side of this slide, where we are able actually to quantify directly on tissue the tumor cells that are carrying this specific marker. And we are also showing that an increased signal of this marker in the tumor cells indicate the payload delivery and DNA damage activity in tissue. So once we have identified the right biological marker, the next question is whether we can measure it in a reliable way. So the second pillar of the SMART framework is measurability. A biomarker only becomes useful if it can measure in a robust and reproducible way. The reliable biomarker require standardized assay workflow, reproducible measurement, quantitative output. I just showed you some cases of quantitative output, and rigorous quality control. And here at Alimi, we are putting a lot of emphasis in terms, of standardizing all our spatial assays at this time to have, this type of critical quantitative and robust output that is necessary for a biomarker readout. And the technology that we are enabling, to do this could be the multiplex immunofluorescence, can be in some regard the imaging mass cytometry, and as well, to some extent, the spatial transcriptomy. Here, an example again of a quantitative output directly on tissue, which is the tumor proliferation marker. We can definitely measure directly on the tissue the Ki-67 that are carried by the tumor cells. So this special technique that we are using here, which is the multiplex immunofluorescence, enable us to measure in a quantitative way the cellular state indirectly on the tissue. And the Ki-67 in that regard quantify the tumor cell proliferation within the tumor compartment. And it enabled, in that particular example, the dose-dependent pharmacodynamic assessment of treatment response. But biological mechanism, it's in tissue often emerge from not only one measure, but emerge, from multiple measure, and spatial capability help us, to reach, that level of complexity in terms, of measuring the different spatial metrics. Here, an example: the cell density, cell proximity, the neighborhood architecture, as well as the colocalization of marker. So measuring this signal allow us actually, to be able to really nail down the mechanistic-driven biomarker strategy to understand the biological coherence of the therapy. But, sorry, the next slide. Measuring this, all these, metrics, it's not, the end of the story. We need to make sure, that we are able to measure these different, metrics in an achievable way. So we need to be able to work within the constraint of the clinical sample during a clinical trial. And those constraints are small biopsies, sometimes a lot of FFPE tissues. If we are looking, at the oncology program, a lot of tumor heterogeneity. So the multiplex assays that we are developing allow us to really measure all the multiple mechanistic signal in the same tissue section, with the bias that we always need actually, to perform an assessment of the quality and robustness within the panel that we are developing. Here you have a panel here, which is composed of CD8, PD-L1, FOXP3, CD68, as well as the pan-cytokeratin marker. But this panel needs to be elevated to some type, of qualification, robust enough to be able to read. So this multiplex approach enables us to capture the spatial biology within limited clinical samples. And with what I showed before around, the elevated spatial metrics that we can grab from just, measuring the marker, we can as well, use, the spatial architecture to predict the response. And here you have an example of non-small cell cancer patients that were treated with a PD-1 inhibitor. In the responder, what you are seeing, it's high CD8 infiltration within the tumor. But a lot as well, on these spatial, very specific metrics that needs to be, quantified as well, which are the CD8 and the tumor interaction. And you see a very short CD8 tumor distance. This is for the responder. In contrary, the non-responder, you have, something which is called a completely immune exclusion phenotype. So the CD8, cells are totally restricted to the stroma and not penetrating the tumor. There is a stromal barrier then preventing the T-cell penetration. So those are the spatial metrics that need as well, that are quantifiable measure of mechanistic-driven objective. So beyond actually, the feasibility, there is something of doing, those assays. Those assays needs to be as well relevant. So the biomarker may also provide the measurement information for drug development. So the mechanism-driven biomarker needs to answer key development questions. Is the drug engaged in this target? For example, what is the target expression for antibody-drug conjugate? Is the pathway being modulated? Do I have a STAT3 signaling suppression? Or which patients are most likely to respond? Immune activation markers such as the CD8 expression or granzyme B. And And eventually, this biomarker need as well, to be optimized across, the whole development. So in preclinical, it needs to confirm the biological mechanism. In early clinical trial, we need to have a readout. They become, a readout of the pharmacodynamic biomarker. And in late clinical trial, they become, the patient stratification. And here again, across, development example of the biomarker continuity within the drug development. So finally, a biomarker to influence decision, the timing, it's really key. And the biomarker will have a greatest impact, when applied early in development. Early mechanistic biomarker will enable the dose selection, will enable the patient stratification, and eventually rational combination strategy. And the late biomarker, something, which is incorporated within, the phase 3 of the program, rarely influence the clinical outcome. However, you can see, from the clinical drug development pipeline, there is, a lot of go and no-go decision that can be taken, if you have real mechanistic strategy for biomarker development. So the question at the end is, how do we integrate all these dimensions into a coherent biomarker strategy? So the modern biomarker strategy will combine the spatial biology, multi-omics profiling, bioanalytical PK/PD measurement, as well as the computational modeling. The goal here is to connect exposure, mechanism, response. And the S.M.A.R.T biomarker strategy that we have showcased today will connect the biological mechanism with clinical decision-making. And the integrated view that you have seen from us today will allow biomarker strategy to move beyond correlation toward causal understanding of treatment response. And with that, I'm going to hand over the final slide to Dr. David Bono for how we're going actually to put in place this biomarker strategy.
Many thanks, Corinne, and thank you very much for these nice examples. So yes, to connect, let's say, exposure, mechanism, and response, what we have done, we have, let's say, consolidated more than 15 years of experience in some ready-to-use panels. So this is something brand new at Aleri, and it took us several months to verify all panels, etc. And our objective is to give access to our partners, clients, sponsors, to give access to some panels of biomarkers that can be directly used on tissue sections, on bulk tissues, for example. So today we have two main collections, one in oncology and the other one in immunology/inflammation. And do not hesitate to scan the QR code on this slide. It will allow you to download the brochures associated to this ready-to-use panel. Why it is interesting? It's interesting because it's a gain of time and money. You can directly have access to some panels of interest, biomarker panels, that can be applied directly on the human tissues of interest without having to wait months of panels development, for example. So if I take the first collection in oncology, you can see here the list of panels. So we are speaking about thousands of markers and the possibility to target transcripts, proteins, metabolites, lipids directly, again, in tissues by preserving the tissue microenvironment if we are using the spatial modalities. And Corinne very well explained why it is a key of success today, more particularly due to the regulatory shift. And we can also use some panels, of course, on fluids, bulk tissues, cell suspension, tissue homogeneity if needed. So you can see here on this list that we can focus directly on some proteins of interest by combining the detection of 20 or more proteins directly on tissue section, depending on your need, depending on the therapeutic indication and what is, let's say, the mechanism of action of the drug more particularly. For the immunology inflammation collection, this is exactly the same. We have the possibility to target the same classes of molecules. We have some dedicated panels like the fibrosis one or the T-cell exhaustion one. So again, these panels are ready to use, can be applied directly on translational clinical projects when working on human tissues. Of course, we need to have access to the tissues, to the biopsies, to be able to apply these panels and deploy, let's say, the smart approach you have seen. So again, feel free to scan the QR code, feel free to go on our website. You can directly download these panels, or feel free to also schedule a meeting with us. So to finish, before answering any question you may have, if I'm really pleased to share an exclusive offer today, so a 5% discount on service fees for any project you may have with us on any biomarker worked you would place with Aleri in 26. So please scan the QR code or visit our website to schedule a meeting, schedule a consultation with Corinne, myself. And if we can have this meeting by March the 18th, you will have access to this exclusive offer. And I really would like to thank you all for joining us today. And I'm extremely happy to answer with Corinne any question you may have. So I invite you to post your question in the Q&A box dedicated to this on the webinar interface. Thank you very much. And I can see we already have some questions. So the first one for you, Corinne. Based on your experience, how important is it today to move toward mechanism-driven biomarker rather than single marker approaches? So it's more a question of simplex versus multiplex approaches. Very good question.
So single biomarker approach, usually we know that efficacy, it's not going, or at least understanding the efficacy of the therapy, it's not going to be explained by only one single biomarker. So you need a context for that. And you need to understand, that context. So in addition of measuring, like multiple biomarker or multiple marker, that will give you like a flavor, actually, like of this context, everything related to spatial biology, molecular interaction between, these different markers, it's going to be really the key of obtaining a real answer around, like the predictivity or the biological mechanism of your therapy.
Thank you. We have another question. So you showed how spatial context can reveal interactions between immune or tumor cells. In practical terms, how does spatial biology improve our ability to predict patient response compared with traditional bulk approaches assays?
So it's the continuity, like of the first question. I mean, the traditional bulk does not measure, like the spatial context, and does not measure, like anything related to what is happening, like really at the site of action. Where do you need to understand, like the mechanism of your therapy? So bulk measurement will never get, like the predictivity of everything related to spatial technique.
Thank you. And another nice question. With the increasing relevance of microbiota in therapies, so microbiotes, are your panel focus only on human-based marker, or do they also include microbiome biomarkers?
So right now they don't. That doesn't mean, that we cannot qualify, like any type, like of biomarker and build, like from scratch, like a panel that it's relevant. And it's exactly what we will be doing. Something, like which is relevant to the mechanism of your disease and the mechanism of the therapy you want to apply to it. So even though, like it's a good start, like the biomarker that we are providing right now in terms, like of panel, we understand, like the opportunity to add, like very specific biomarker related to your biological mechanism. So it's an effort from us and something, that we will qualify, like, as I say, in a very robust manner that will help us, like to really accurately depict, like the predictability of your therapy within the tissue.
Absolutely. So we have these ready-to-use panels available, but we can also personalize, customize any biomarker of panels of interest. So feel free to contact us for this type of purpose. Next question. Why do you choose one platform, I assume a spatial platform, over another for your projects?
You mean from one special platform to another special
Yeah,
platform?
why selecting this specific platform, for example, the GeoMX
No, to
compared to
tell
the
you the truth,
MC and
right
vice
now,
versa?
I want, like in spatial capability, what I'm looking for, it's really something that can resolve, like the signal at the single cell. So this is, like one of the way, like I say, I will really choose, like the platform. Can I get a reading, like at this subcellular resolution? Because this is exactly, the way our biomarker framework is working. We're looking at signaling pathways, so I need to understand, like what is happening within the cell, but at the microenvironment, and as I said, like within the tissue complexity and within, like the abundance of the different cells. So one of my preference right now, I would say one of the strategies that we have, it's really, like to get, like a cell resolution readout, like with our special platform.
Thank you. Another nice question. Yeah. You emphasize the importance of biological context and tissue level understanding when building biomarker strategies. How do you see newer models like patient-derived organoids contributing to biomarker discovery compared with traditional preclinical systems such as mouse models? Very hot topics, more particularly following
Very
the
hot topic, and
announcement of
this
the
is also,
FDA last year.
like something which we are working, like actively as well. At least, like from a research and development purpose right now, like at Tannery, but eventually, like this is something we're going to propose, like to our sponsor. Great. I mean, we can work actually, like with this organoid and help us, like to understand from a mechanistic, like point of view without wasting, like too many animals, what it's really relevant because we can play using, like this model organoid or something, that we're trying to put in place. It's what we call a human NAMS right now, but we try actually, like to understand the different layer, and they help us to understand the different layer of biological complexity that we need to understand. As I said, like the target engagement, the signaling pathway, the microenvironment. So no, I mean, it's great.
Thank you. Next question. What platform is used for lipidomics and proteomics? Are they validated already?
I don't know which platform you're talking about.
I assume people are really asking just a technical platform we are using for. So for lipidomics, we are using the mass spec imaging platform.
So right now,
So
this one, like it's not validated to some type of GCP compliance. So we have validated everything related to proteomics and to a certain extent, like transcriptomic as well. So right now, we are pleased, like to offer this type of platform in the GCP environment in our company.
and for the proteomics part, we are using the classical immunostaining, immunohistochemistry approaches, as well as the GeoMX. For example, we can go up to 1,200 proteins, if I'm correct, directly detected directly in tissues. Next question, are we developing NAMS approaches?
So yes, the NAMS approach, yes. We are. This is something that we're working actively, like in the research and development unit that I'm directing right now. Not to the point where we can offer that services, like to our clients so far, but we're working actively, like to be able to provide that within, like the next half of this year. The second half of this year. Sorry about this.
Thanks. Thank you very much, Corinne. And I think I have covered the majority of the question. We have also got people asking about how much Valérie has experience working with S.M.A.R.T samples. So I have an example. It's plasma-derived samples or point-of-care tests.
So we don't have experience with the point of care test for sure. We do have some, so that means, like maybe, I guess, like when you're saying point of care test is very small amount, like probably of blood or something like this. So we don't have expertise with this. We are working, like in sometimes, like a biomarker strategy with fluid as well, but more related to serum or CNS, for example, fluid.
Perfect. I don't see. Thank you very much for all your questions. Much appreciated. We can see that it is a very hot topic. Are those, last question, are those biomarker assays or clear assays?
So
A
you
good question.
understood that this is the next step. So right
Yeah.
now we are at the point where we qualify, like those biomarker panel to reach, like a certain level of pharmacodynamic and efficacy, like endpoint that we want to assure, like in clinical trial. Eventually, when we will move, like to stratification, we'll have to build, like some clear.
Thank you, Corinne. It was the last question in the Q&A box. So thank you very much, everyone, for
Thank you.
joining us. The webinar has been recorded, so it will be possible to have access to the records. Feel really free to contact us directly on our website. You can schedule a meeting by directly submitting your requests. You can scan the QR code here if needed as well. And yeah, looking forward to answering any other question you may have during some meeting with you. So thank you very much. Have a nice day. Bye bye.
Thank you.